Hello everyone, this is second part of the lesson Structural and Functional Unit of Life. In this part we will discuss about cell organelles, their types, cell inclusion, mitosis and meiosis, importance of osmosis and diffusion, protoplasm theory, centrosome and centriole, the plants and animal cell, stem cell and types of chromosome. We are going to start. The first one is the cell organelles. Cell organelles means tiny organs or little organs. Little in the sense that they are the parts of the cell, or we can say microscopic structure that are found inside the cell. These structures are embedded in cytoplasm and they perform different metabolic functions such as nutrition, respiration, transportation of materials, excretion, etc. These organelles do the same work as organs do in entire body of plants or animals. That's why the name organelles given to it. Cell organelles माइक्रोस्कोपिक स्ट्रक्चर हैं जो सेल के अंदर में पाए जाएंगे. ये साइटोप्लाज्म में एम्बेडेड होते हैं. इनको सेल ऑर्गेनेल्स इसलिए कहते हैं क्योंकि जिस तरह से हमारी बॉडी में ऑर्गन वर्क करते हैं, ऐसे ही सेल के अंदर सेल भी लिविंग थिंग्स है और सेल के अंदर बहुत सारी एक्टिविटीज़ चलती रहती है, जिसको ये सारे ऑर्गेनेल, ये सारे माइक्रोस्कोपिक लिटिल स्ट्रक्चर परफॉर्म करते हैं. इसीलिए इनको हम सेल ऑर्गेनेल्स कहते हैं. This one is the first cell organelle in this slide, that is the endoplasmic reticulum. It is the network of tube-like structure that is running all through the cytoplasm. We can say that endoplasmic reticulum forms a channel through which required material transported in the cell. If we observe the endoplasmic reticulum, we find that outer end of endoplasmic reticulum connected with the cell membrane and inner end of endoplasmic reticulum is connected with the nuclear membrane. And this organelle cover the near about all the spaces, we can say entire cytoplasm in a cell. When ribosome are attached with the endoplasmic reticulum, that is known as rough endoplasmic reticulum or RER, and when ribosome is not attached with the endoplasmic reticulum, then that is considered as smooth endoplasmic reticulum. Overall function of endoplasmic reticulum is formation of protein and lipid, and because it covers the entire cytoplasm, that's why increase the surface area of the cytoplasm. And smooth endoplasmic reticulum helps in the detoxifying many harmful substances inside the cell. It also helps in the constituting the cell membrane by the process of membrane biogenesis. इंडोप्लाज्मिक रेटिकुलम ऑर्गेनेल है जो लगभग कंप्लीटली पूरे साइटोप्लाज्म में स्प्रेड होता है, फैला रहता है। इंडोप्लाज्मिक रेटिकुलम का जो आउटर एंड है वो सेल मेम्ब्रेन से जुड़ा होता है, जबकि इसका इनर एंड न्यूक्लियर मेम्ब्रेन से जुड़ा होता है। इंडोप्लाज्मिक रेटिकुलम को दो भाग में क्लासिफाई हम लोग कर सकते हैं, एक RER रफ इंडोप्लाज्मिक रेटिकुलम, दूसरा स्मूथ इंडोप्लाज्मिक रेटिकुलम। इंडोप्लाज्मिक रेटिकुलम में अगर राइबोसोम अटैच है तो उसको हम RER बोलेंगे, जिसका फंक्शन होगा प्रोटीन सिंथेसिस। और अगर राइबोसोम ER से अटैच नहीं है तो वो बनाएगा स्मूथ इंडोप्लाज्मिक रेटिकुलम, जिसका फंक्शन है लिपिड सिंथेसिस। बिकॉज the cell membrane is made up of lipid and protein, and the same things endoplasmic reticulum is also producing. So ER has a role in constitution of cell membrane, and the process is known as membrane biogenesis. SCR, अगर सेल में कोई पॉइजनस सब्सटेंस या हार्मफुल कंपोनेंट बन रहे हैं तो उसको डिटॉक्सिफाई करने में भी हेल्प करेगा। और ओवरऑल इफ़ वी आर टॉकिंग अबाउट द इंडोप्लाज्मिक रेटिकुलम, देन इंडोप्लाज्मिक रेटिकुलम साइटोप्लाज्म का सरफेस एरिया बढ़ाता है, क्योंकि ये लगभग एंटायर साइटोप्लाज्म में फैला हुआ रहता है और एक ट्रांसपोर्टिंग चैनल की तरह से वर्क करता है, जिसके द्वारा सेल के रिक्वायर्ड मटेरियल सेल के अंदर में डिफरेंट ऑर्गेनेल को ट्रांसपोर्ट होता है। नेक्स्ट इज राइबोसोम। दिस इज द साइट ऑफ प्रोटीन सिंथेसिस। दिस वन इज द स्मॉलेस्ट ऑर्गेनेल, and ribosomes are the organelle that is produced by nucleoplasm. In previous video it was explained to you the role of nucleolus. Ribosome is the structure organelle that either found attached with the endoplasmic reticulum or found free inside the cytoplasm. There are two types on the basis of size. A ribosome can be classified as 70S and 80S. 70S ribosomes are found in prokaryotic cell, whereas 80S ribosomes are found in eukaryotic cell. But the function is common. It helps in the it works to synthesize protein. Next is mitochondria. This is the small rod-shaped organelle that is found in large number. If we compare plant cell and animal cell, then animal cell required more energy. That's why more number of mitochondria are found in the animal cell with the compare of plant cell. Outer membrane, mitochondria is bounded by two membranes, that is the outer and inner. And the outer membrane is smooth, whereas the inner membrane is pushed inwards and from cristae. Mitochondria is the cell organelle where cellular respiration takes place. Respiration means oxidation of food to produce energy. So this is the site where energy is released in the form of ATP. That's why it is known as powerhouse of the cell. One more thing about the mitochondria is that it have their own DNA and ribosome, so they can synthesize their own protein, and that's why they are the semi-autonomous organelle. The next one is the Golgi bodies. In plant cell, Golgi bodies is known as dictyosomes. This organelle was discovered by Camillo Golgi, and it is formed by stacks of flat membrane or flat sac called cisternae. Some vacuoles may attach with the cisternae, the sac-like structure. The function of Golgi bodies is to store, modify, package, etc. Lysosomes are also found by Golgi bodies. Acrosome, that is the part of human sperm, also found by Golgi bodies. It is found near the endoplasmic reticulum and nucleus inside the cell. Next one is the lysosome. This is the organelle which help to keep the cell clean, and it is produced by Golgi bodies. It is told to you earlier. Lysosome, the organelle which are filled with digestive enzyme. When any cell organelle are misbehaving or not functioning well, lysosome has ability or capacity to digest them. Sometimes lysosome digest themselves also while engulfing that particular cell in which ribosome is lying. Sometimes lysosome digest their own cell. That's why it is also known as suicidal bag of the cell. Next is the plastids, largest organelle in the cell, and you are familiar with the plastids such as chloroplast. As you know that chloroplast means green colored plastids, and from lower standard students studying about the chlorophyll, the pigment which trap the sunlight and help the plant to begin the process of photosynthesis. So chloroplast is common plastids. If we see the other type of plastid, then leucoplast and chromoplast are also. The first one is the leucoplast, that is the colorless plastids. Means no pigments are found in these plastids. The function of these plastids is to store food such as starch. Common example is potato, which are having lot of leucoplast in them. Other example may be just like garlic. Edible root or stem that is found under the soil contain large number of leucoplast. Next one is the chromoplast. Chromoplast are the colored plastids. They are responsible for different colors of the fruits, flowers, etc. There are different types of chromoplast found. In other words, we can say chloroplast is a type of chromoplast because chloroplast indicate the green colored plastids. There are different colored plastids are available, such as yellow colored plastids are known as xanthophyll, orange red colored plastids are known as carotene, and just like that different colored plastids have different names. Some coloring pigments such as blue, violet, purple are not associated with plastids. because these colors almost dissolved in the cell sap and give that color to the plant structure, and such pigment are called anthocyanin. Example is croton plant, where we can see different colored pigment. Chloroplast, as we are discussing earlier, that this one is the plastid which helps in the photosynthesis because it stores green colored pigment called chlorophyll, and chlorophyll having ability that to hold the sunlight to begin the process of photosynthesis. During the photosynthesis, plants produce starch, or we can say glucose, and oxygen. Glucose is utilized by the plants to follow growth and development, whereas oxygen is utilized by the plant to follow respiration, and plants and animal both. Plastids also have their own genetic material, that's why plastids have ability to divide itself. This is also semi-autonomous organelle. And the next one is the cell inclusion. Cell inclusion means non-living parts are the components of the cell, in which it includes number of food materials such as starch, glycogen, fats, amino acid, etc. Cell inclusion can be classified into two, that is the granules and vacuoles. Granules are the small particles like structure that is embedded in the cytoplasm. These are starch, glycogen, fats, amino acid, etc. And vacuoles are membrane bound structure, sac like structure, which contains solid as well as liquid materials. Vacuoles are of three types. First one is the sap vacuole, second is food vacuoles, and third is contractile vacuole. Sap vacuole are filled with the liquid content in plant as well as animal cell. In plant cell, this vacuole cover a large volume. Sometimes vacuole occupy 50 to 90% of the cell volume. Outer covering of sap vacuole is known as tonoplast. Cell sap is means liquid content of the cell, or we can say membrane bound organelle which is filled with the liquid content, that is the cell sap. If you want to understand sap means, then you have seen immature wheat that is green in color. If we take it in hands and push it, then you will see the white milky color appearance comes out, that is the wheat sap. देखे होंगे आप, जब गेहूँ मैच्योर हो रहा होता है तो हरा होता है, उसका सीड हरा होता है. आप उस सीड को अगर हाथ में लेके उसको दबाएंगे तो उससे मिल्की वाइट लिक्विड बाहर आएगा, उसको हम व्हीट सैप बोल सकते हैं. तो सैप मीन्स लिक्विड कंटेंट. सैप को समझाने के लिए आपको एग्जांपल दिया गया. This sap vacuole may store salt, sugar, amino acid, organic acid, and some other proteins. Sometimes these vacuoles may store waste material. Sometimes they also store enzymes. And these vacuoles helps in maintaining turbidity, flaccidity, and rigidity of the cell. And these are also required for osmotic absorption of water. Next one is the food vacuole. You studied in the class 7 that food vacuoles are found in the amoeba or the amoeboid cell of higher animals. You studied that amoeba has pseudopodia false feet, which helps in the holding submicroscopic food of amoeba. Pseudopodia fuse together to form food vacuole, and inside the food vacuole the food particle is digested. due to action of enzymes released by the food vacuoles. So that is the food vacuole found in amoeba, etc. Contractile vacuole. These are the vacuole which are found in the fresh water organisms such as amoeba, paramecium, chlamydomonas. So amoeba and paramecium are protozoans, whereas the chlamydomonas is the unicellular alga. And these vacuoles having ability to collect liquid from the cell, swell up, and sometimes they burst also to release their contents. These vacuoles helps in the regulation of water and maintaining the quantity of water inside the cell. They also helps in excretion, removal of the waste material from the cell. This one is the important topic, that types of cell division in the cell. So there are two types of cell division. First one is the mitosis and second is meiosis. So mitosis is just a type of cell division that result in the formation of two daughter cells. Means when a cell divides to form the new cell, they have to follow a type of cell division, that is the mitosis, where two daughter cell or the two new cell are formed, and each cell has the same number and kind of chromosome as the parent cell. This is the fundamental process of life. Mitotic division always the equational division, because number of chromosomes remains the same. During this type of cell division, means during mitotic division, cell copy all its genetic material and split into two identical daughter cells. Mitosis has some advantages as well as some disadvantages also. Advantages means they take less time to follow division, but disadvantage is because exact copying is going on. That's why if any disease or other factor are also there, then that is also going to copy in the next new cells. Phases of mitotic division is the prophase, metaphase, anaphase and telophase. In short, you may write PMAT, PMAT. Result of mitotic cell division is two nuclei that are identical to the parent nucleus. Mitotic division takes place in the somatic cell. All human cells are somatic cell except sex cell, means male gamete and female gamete को अगर छोड़ दिया जाए तो बाकी सारे सेल्स जो ह्यूमन सेल हैं वो सब सोमेटिक सेल हैं, और सोमेटिक सेल में माइटोटिक डिवीजन ही पाया जाता है. इसको एग्जांपल हम ले सकते हैं, जैसे कभी कभी हम लोगों को समटाइम वी हर्ट आवर सेल्फ, कभी कभी हम सबको चोट लग जाती है जिसमें वुंड्स वगैरह भी हो जाते हैं. तो वुंड्स की हीलिंग में जो सेल डिवीज़न होता है वो माइटोटिक डिवीज़न है. सोमेटिक सेल डिप्लॉयड होते हैं. डिप्लॉयड मीन्स दे हैव 23 पेयर ऑफ क्रोमोसोम and means 2 copies of each chromosome, whereas the gametes are haploid as they have 1 copy of each chromosome, means 23 chromosome. Gametes are haploid in the sense that they have only 23 chromosome, whereas somatic cell are diploid in the sense they has 23 pairs of chromosome. We are moving for the next and going to talk about meiosis. It is a type of cell division that is known as reductional division, because the number of chromosome reduced to half. It is differ from mitosis in reducing the number of chromosome taking part in the process, hence this one is known as reductional division. It is a two-step process that includes the reduction of the chromosome number by half, as it is told to you in the previous slide that male and female gamete are haploid. When male and female gamete unite together and form a single diploid cell, that means a 2n cell, then they acquire the 23 pair or 46 chromosome. So half number of chromosomes that are formed during meiosis is compensated after fertilization. When male gamete and female gamete both unite together, then they achieve the number of chromosome exact it is, that is the 46, and this is the actual number of chromosome acquired by embryo. Meiosis has two phases, meiosis 1 and meiosis 2, and this topic is broad in the sense that number of phases are there, so it will take more time to explain. So I think separate video should be there for the mitosis and meiosis. So next video can be in coming future for mitosis and meiosis. We are moving towards next. So importance of osmosis and diffusion. If we want to define osmosis and diffusion, then the common definition is movement of substances or the molecules from higher concentration to lower concentration. And diffusion apply to the activity of many types of molecules, but when we use osmosis, then this is the movement of only water or the liquid content. And both of the movement, or we can say diffusion and osmosis, both are the example of passive transport because they do not require energy. These are important in the sense because number of unicellular organisms which follow respiration follow the diffusion to satisfy their oxygen need. Single celled organism that live in fresh water, they gain water by the process of osmosis. Next is plant are also the example where root absorb water through this process, that is osmosis. Osmosis also important in the sense that opening and closing of stomata is also governed by the process, that is osmosis, where gain and loss of water through which guard cell open and close the stomata is also governed by osmosis. So these are the few importance of osmosis. And the next one is the water that is absorbed by the roots of the plant. There is also osmosis. Xylem vessels which transport the water to different parts of the plant, there is also osmosis. And food that is produced by the phloem tissue, food synthesized by the leaves, that is transported by the phloem tissue, there is also involvement of the principle of osmosis. In sieve tubes also this phenomenon, or we can say principle of osmosis, are used. So with this example we can understand that both of these processes are important in this environment. Next is the protoplasm theory, and as you studied the cell theory suggests that all living organisms are made up of cells, and the protoplasm theory suggests that all living organisms are made up of protoplasm. Protoplasm is the structure that is made up of cytoplasm and nucleus. It is proposed by Max Schultz, and later O. Hartwig was the man who proposed that the protoplasm is a series of fiber that is responsible for the movement of the cell. Protoplasm, if we want to define it, this is also semi-fluid like structure which is made up of carbohydrate, fat, proteins, vitamins and minerals, and we can say all the semi-liquid like material from cytoplasm, from cell membrane to the nucleus hold together semi-fluid like structure known as protoplasm, that is including cytoplasm as well as nucleus. Next is the centrosome, and centrosome is the distinct region of the cytoplasm near to the nucleus in animal cell, and it has two central granules called centrioles. Centrioles are hollow cylindrical structure made of microtubules that is arranged in a specific manner, and these two centrioles are arranged at right angle to each other. The function of centrosome is to maintain equal distribution of chromosome in daughter cells, and they also help in the formation of cilia and flagella. Next is these are the slides which indicate the differences between plant cell and animal cell, and you know that outermost covering of the plant cell is the cell wall, whereas the outermost covering of animal cell is the cell membrane, and in plant cell vacuole is the large sized organelle that cover the maximum volume of the plant cell. In plant cell plastids is found, in animal cell plastids are absent, and other organelle are common in both. One more thing we can consider, that the Golgi complex name of Golgi complex is dictyosomes in plant cell. Centrioles are present in the animal cell, absent in the plant cell. Differences between plant cell and animal cell. You may read these differences, and these are very common to you people, so we are moving towards the next. Some differences you may look this as plant cell store food in the form of starch, whereas the animal cell store food in the form of glycogen, and other are common, you studied the same. Going for the next, that is the stem cell, and this is the important topic for medical science, as these cells having ability to develop into specialized cell types in the body. These type of cell having great importance in the sense that they are helping the doctors and other people who are involving themselves in many research. They are able to understand the basic biology of how living things work, and what happens in different types of cell during diseases, and stem cells therapy, or we can say stem cells research, also help to replace lost or damaged cell that our body cannot replace naturally. Means if medical science achieved uh, such things, that how to regenerate the lost cell or the tissue or the organ, then it will be the big achievement for the medical science. So here our body is made up of different types of cell, and most cells are specialized to perform particular functions, such as the RBC having the ability to carry oxygen around our bodies, but RBC are unable to divide. So stem cell can provide new cell for the body as it grows, and replace the specialized cell that are damaged or lost. Stem cell having unique properties, such as they can divide over and over again to produce new cell, and as they divide they can change into other types of cell that make up the body. There are three types of stem cell. One is embryonic stem cell, second is adult stem cell, and third is induced pluripotent stem cell. The embryonic stem cell is the stem cell that supply new cell for an embryo as embryo grow and develop into a baby. So this is the stem cell which is a boon for those parents who does not have baby. Next is the adult stem cell. Adult stem cell supply the new cell as an organism grow and replace cells that get damaged. Adult stem cell are the multipotent, as they can change into some cells in the body, but not any cell. For example, blood stem cell replace the blood cell, and skin stem cell helps in the replacement of the skin and hair, because skin and hair both are attached to each other. So adult stem cell can helps in regeneration of blood stem will regenerate blood cell, and skin stem cell will regenerate the skin and hair. Next one is the induced pluripotent cell. These are the stem cell which are also known as iPS cell, and these cells are produced by scientists in the laboratory. Induced means because they are produced, again they are produced not by the natural means, by the human, that's why they are the induced. Like skin are the blood cells, and reprogramming them to become stem cells. Just like that, embryonic stem cell, these are also pluripotent, so that they can develop into any cell type. So usefulness of stem cell is to, in research, it is, as I told to you, that how the different type of cell behave during the diseases, and how to regenerate, or we can say replace, the lost or damaged cell in our body. So these are the usefulness of stem cell. And totipotent cells are there that having ability to regenerate. If medical science achieves such things, then it will be a great achievement. Next, this one is a quick recap. Plasma membrane, you know very well that this is the layer which separates cell from external environment. Cytoplasm, you know that this is the semi-fluid like material which are found from cell membrane to nuclear membrane. and this is the nutritive medium of the cell. Nucleus is the control center of the cell, which has their own DNA, and there is also ribosome formation by the nucleolus. Ribosome is the protein synthesis sites. Mitochondria is the site where ATP energy is produced in the form of ATP. Peroxisome are the organelles which are found near ER, and these are the organelles which oxidize and break down fatty acid and amino acid and detoxify poisons. Vesicles and vacuoles are helps in storage and transport. Centrosome is the this is the organelle which are found near to the nucleus in animal cell, source of microtubule, and helps in the equational division of chromosome. Lysosomes are organelle which keep the cell clean, filled with the enzymes. This is also known as suicidal bag of the cell. Cell wall is the protectional or the structural support, or we can say outermost covering of the plant cell, which give the permanent shape to the plant cell. And the chloroplast helps in the photosynthesis. This is the plastid, green color plastids. Endoplasmic reticulum is the channel-like structure which transport the different required material to every parts of the cell. It forms protein and lipids and help in the membrane biogenesis. Golgi apparatus is the storehouse of the cell, as it store different required material, helps in the packaging and modification of the complex food. Cytoskeleton that is the help in the maintenance of the cell shape. Sometimes ER also considered as cytoskeletal framework of the cell. Flagella is the cilia and flagella are the structure that helps in the locomotion. And the next one is the types of chromosome. This one will be last topic of this part, second part. In this section we will discuss about different types of chromosome. So discovery of chromosome was in 1842. Carl Nageli was the first person who has observed the rod-like structure present in the plant cell nucleus, and it is generally known that chromosomes were first discovered by Walter Fleming in 1882. In 1888, von Waldeyer Hertz coined the term chromosome. But in 1902, the two scientists Theodore Boveri and Walter Sutton, they worked separately and found that chromosomes are the carrier of genetic material or genes. Means from 1842 to 1902, it longer duration of the time it took to define that the chromosome are the material which transfer character from parent to offspring. Except of these, chromosome varies from species to species. As bacteria, if we are talking about bacteria, then they have the circular chromosome, whereas plant, animal, human have the linear chromosome. And the number of chromosomes vary from species to species. As you studied that animals like human have 23 pair of chromosome, and out of 23 pair, 22 pair is autosomes and 1 pair is the sex chromosome. The fruit fly has the 4 pair of chromosome, rice plant has 12 pair of chromosome, and dog has 39 pair of chromosome. In this way you can think that every living organism has different number of chromosomes, but you also know that sometimes same number of chromosome are found in the different species. Chromosome is constituent of the chromosome is it is made up of histone protein and DNA, and that is having the three-dimensional structure that is known as double helix. In the previous slides where images you observed that there were structure of chromosome are shown. You may see from there that the types of chromosome, the shapes of chromosome. So if we are talking about human chromosome, the 46 chromosomes are found in the human cell, but they all are not exactly the same. So they are different type based on their shape, structure and function. Chromosome types based on the number of centromere present on a chromosome. Here are different types of chromosome that is classified on the basis of number of centromere present on chromosome. First one is the monocentric. Monocentric, if chromosome has only one centromere. Mono means one and di means two. If chromosome has two centromere, then that is dicentric. And polycentric if chromosome has more than two centromere. Acentric if chromosome doesn't have any centromere. And these are acentric chromosomes, or we can say that these are the broken segment of chromosome that do not survive for long. Sometimes chromosome do not survive for longer duration of the time. So broken part of that chromosomes are known as acentric. Diffused or non-located chromosomes are those which has doesn't have distinct centromere, means its centromere is diffused throughout the length of the chromosome, means not having exact position where the other centromere are found. So diffused, they are the diffused chromosome. Next is the chromosome type based on the location of centromere. So on the basis of location of centromere, that they are classified into four. First one, the metacentric. In this type of chromosome, the centromere is located almost at the center of the chromosome, and due to which two arms, that is P arms and Q arms of the chromosome are seems to have equal length and form V-shaped structure. Such human chromosome are metacentric chromosome, and chromosome number 1, 3, 16, 19 and 20 are metacentric. Submetacentric chromosome are those chromosomes which are in which centromere is located slightly away from the midpoint, and that's why P arm is shorter than the Q arm, and due to this position of the centromere, chromosome acquire L shape, and chromosome number 2, 4 to 12, 17 and 18 and X chromosome are submetacentric chromosome. Acrocentric chromosome are those which are found far from the center. We can say these centromere in this type of chromosome, acrocentric chromosome, the centromere is located near to the end region and take the subterminal position, and due to that P arm is shorter and Q arm is longer, and these chromosomes are 13 number, 14, 15, 21, 22 and Y. These are the acrocentric chromosome. The last one is the telocentric chromosome. It is the rod shaped chromosome in which centromere is located at the proximal end, means the tip of the chromosome. Hence P arm does not exist. This type of chromosome is not found in the human. This is the telocentric. Finally, we reach to the types of chromosome on the basis of their function. So there are two types of chromosome in human chromosome. The first one is the autosomes and second is the sex chromosome. In class 8 you studied that the 22 pair of chromosome which are responsible for carrying the different characters from parent to offspring, except identification of the sex, whether the child is going to be boy or girls. So except identification of the sex, the child is going to have all other character are carried by autosomes from parent to offspring, and autosomes are the same for both male and female. The two chromosomes form the homologous pair, means carry the same character, and the sex chromosome, the last pair of this chromosome, 23rd pair of the chromosome, the last pair, which helps in determining of the sex of the individual, that is the sex chromosome. You studied in class 8 that if identical pair of the chromosome, that is the XX, the individual will be female, and if non-identical, means XY chromosome, then the individual will be male. So sex chromosome are not the same for the male and female, that is X and XY, and here are another name for sex chromosome, that is the allosomes. So this is all about the types of chromosome. Thank you for watching this video. Study, in my opinion, is not easy. That first of all, you will have to learn how to spend time with the subject, and when you spend time with the subject, you will slowly begin to understand them, and result will be better. So keep studying and keep learning. We will meet again in the next video. Thank you.